We are getting back to what we started talking about before we all departed and our brains departed and our bodies departed. Genetic mapping. So this is a quick couple slide review of what we talked about a week and a half ago. And the idea here is that one of the main goals of geneticists is to try to figure out what genes cause particular traits. This is especially important for finding genes that cause diseases. So how do we do that? This is a simple example we went through last class, 2 weeks ago, where you can imagine there's a single allele, a big A, and for every big A allele you have, your skin cells make a little bit more pigment. So you have three situations. You have the situation where you have two big A alleles, each one denoted here by a single black dot, so diploid. Two big A alleles, your skin cells make twice as much melanin, dark pigment, as an individual who's a heterozygote, who's an intermediate skin tone, compared to a homozygote with two little a's, whose skin cells make very little or no melanin, pigment. So how would we go about figuring out what gene causes such a simple trait, skin color? This is simple mapping. This is where we think that there's a single gene that controls this trait, two different alleles. And we went through this exercise last time we had a lecture. And the idea here, we did this. So which gene, is it B, C, D, E, or F, that seems to control the trait of pigmentation? And the way that you do this sort of genetic mapping, as a reminder, is you look for perfect correlations between the phenotype and the genotype. So we have 8 different individuals. For each individual, we've got the genotypes on 2 different chromosomes, shown up here at the top, genotypes. So 2 alleles at each gene, and we've got their phenotype. So we look, for example, the best way to start is to focus on every individual that's got the same phenotype and ask what's similar about their genetics. In this case, we found that both of those individuals that had the darkest skin shared the presence of 2 big F alleles at this gene. Then we'd ask something like, is that consistent with the genotypes of the rest of the individuals? So for example, do all of the individuals that are little f over little f have the lightest skin color? Once again, there's a perfect correlation between the genotype of these individuals and their phenotype, the trait. And then all of the intermediates, the heterozygotes, have intermediate skin tones. So this is sort of the ideal situation for genetic mapping. We've identified that it seems like gene F controls this phenotype. The genotype that you have at this gene, are you homozygous or heterozygous, is associated with the trait that you display. Then we've got polygenic traits, which are most of the traits that you and I are probably interested in any organism, plant, animal, bacterium. Most of the traits, human height, for example, are controlled by multiple genes and also by the environment. So it's how much you eat, it's where you grew up, plus it's probably the action of 5, 10, 15, 20 different genes all across your genome. So how would you go about figuring out the complex combination of genes that all together add up to a phenotype that's not discrete or binary like the skin tone situation? We've got light, medium, dark. This is a continuous trait. So complex traits are also continuous. They vary. There's no binary. It's not either you have the trait or you don't. It's not simple Mendelian genetics where you expect homozygotes, heterozygotes, and homozygotes. So there might be 3 different versions of the trait. We don't have short humans, medium humans, and tall humans, in other words, in a 1 to 2 to 1 ratio. We have this sort of variation. So I'm just going to really briefly, before we get into brand new topics, explain how geneticists go about figuring out what genes are associated with complex traits. Using exactly the same example, skin color, just a little bit more complicated. So in this example from a textbook, not your PDF textbook, they imagine 3 different genes, gene A, B, and C. But it's the exact same scenario. Each of those capital letters produces a little bit more pigment. So if you have a capital letter at A or B or C, your cells make a little bit more pigment. And there's only one combination down here in the bottom that gives you 6 out of 6 capital letters. So that's the darkest skin color that could be produced. On the other end of the spectrum, you have 0 of the 6 alleles are capital letters. That's the lightest skin tone that could be produced in this example. So instead of, because there are 3 different genes and 6 different alleles then, big A, little a, big B, little b, big C, little c, this is definitely not binary. It's not light skin, dark skin. It's not even Mendelian. It's not a 1 to 2 to 1 ratio of light to medium to dark skin. It's this. This is the expected frequency of different skin colors you'd expect. And the more genes that contribute to a trait, the more bell-shaped that distribution gets. So you expect individuals of pretty much every skin color, continuous, just like height. So individuals that have say 6 out of 7, there are 15 out of 64 individuals that have 6 out of 7, 5 out of 7 rather, capital letters. So there's this continuous variation in the frequency and in the pigmentation that's produced, depending on how many capital letters you have in your genotype. So then how would geneticists go about figuring out which genes are involved in these traits? It's a little bit more complicated than simple looking for correlations between genotype and phenotype. This is complex trait mapping. Do this in about 5 minutes or so. So the only thing that I want you to take away from this slide, bless you, is just the approach. I won't be asking you to do this per se on any exams. I just want you to understand how much more complicated complex mapping is. So in this example, there are 2 different genotypes, red and blue, and there are 2 different traits. Leaves have a lot of trichomes or a few trichomes, hairs. Okay. So you cross them together, you get an intermediate, genetically intermediate F1, 1 red chromosome, 1 blue chromosome, sure, and also an intermediate trait, kind of partly hairy, half hairy, and whatever you want to call it. So start with simple genetic mapping. If you look in the this says F8. It's an advanced generation. 8 generations into crossing siblings together. You generate individuals with these different chromosomal genotypes. So you've got mixtures of red and blue throughout the genome. And each of these individuals has one of the 2 parental phenotypes. They're either hairy or they're not so hairy, leaved. What gene controls this trait? What part of each chromosome perfectly correlates with the trait? So let's focus on the 4 individuals that are the least hairy. Is there something about their genotypes, that is the color on one part of their chromosome, that perfectly correlates with lack of hairs? Is there any one spot on this chromosome where all of those 4 individuals share the same genotype? What color? Red. On the bottom, red. So there's a spot here where every one of those individuals has red. So that's a good correlation. So it seems like maybe there's a gene in there where if you get, oh, by the way, parents, red allele, red color, means many hairs or few hairs? Few hairs. So that's a good correlation. You've got the red parent's version of that allele, that gene, you get few hairs. So far this seems to make sense. What's the problem? Why isn't this yet a great explanation for what genes cause this trait? So it's true that all 4 of those individuals share red alleles at that spot on that chromosome. Do other individuals also have red there? Yeah, so how do you explain then that this individual has red but has lots of hairs on the leaves? This is where this is why it's complex. So you look for an association that seems pretty good. This is a pretty good association. Most of the individuals that have few hairs have red in this part of the chromosome. But there are some issues too, like we just pointed out. So let's assume, instead of just looking for one gene, let's assume we're looking for 2 genes at 2 different parts of the chromosome. Together, that pair of genotypes controls the trait. So instead of thinking about 1 gene controls a trait, let's think about 2 genes controls a trait. So for each of the individuals that have few leaves, is there another part of their chromosome that also is consistently red? So we'll call this locus 1, gene 1.
Just a little higher up.
So we've got that little block of chromosome 2, is also consistently red in every individual that has few leaves. Is there any plant that is hairy that's red at gene 1, red at gene 2? So it looks like maybe, I don't know, is this plant? It's pretty close. Hard to tell. But this is how complex genetic mapping works. So you add more and more regions of chromosomes and you ask, when you get a complex mixture of genes, do they seem to control the trait? And this is exactly what happens. So we've got one locus here where most of the individuals that have red have few hairs on the leaves. If you add a second locus a little bit farther down, you can see on the bottom, I've summarized the phenotypes of these individuals down here and their genotypes. So every individual that is hairless or relatively less hairy is red at gene 1 and red at gene 2. And the same is true for that leaf, and that leaf, and that leaf, those plants. So this now seems to be a 2-gene trait. That's what these results suggest. You have to have the red allele at gene 1 and the red allele at gene 2 to get a hairless or relatively less hairy leaf. So which one's recessive, red or blue?
Red.
How would you know that?
There's blue, there's more hair.
So when you've got a heterozygote, what's the phenotype?
Very hairy.
It's the same, very hairy. Yeah, it's the same as the blue homozygote. So that suggests that red is recessive. Maybe. Suggests, doesn't prove. So it would be a hypothesis to do further testing on, to inquire about. So that's the concept of complex mapping. Instead of looking for 1 gene controls the trait, you look for 2 genes or 3 genes or 4 genes all across the genome together. There's a combination of alleles perfectly correlate with the trait that you're studying. Now, I promised you, is there any questions, by the way? Yeah, please.
I'm confused on the F1
Yep.
configuration. Are we supposed to just look at the left allele? Or I see red and blue.
Right, so the F1, in this case, they're suggesting that the F1, for example, both of those loci are heterozygous, so it has an intermediate phenotype. That was where this exercise was started anyway, was they wanted to say that the F1 has an intermediate phenotype, But when you break apart, so this is confusing because there's a diploid, so you can be heterozygote. Heterozygous is an F1. But by the F8 generation, they're saying each of these individuals is homozygous. That is, what they're not showing you for each of these individuals is there's another chromosome for each of these individuals, but it looks exactly identical to the one that they've already drawn. Like this. If you'll take my choice of colors a little bit liberally. Yeah. So a heterozygote for a gene would have an intermediate phenotype, but when you get down to this F8 generation, every individual is homozygous at every gene. Questions, other questions, concerns, follow-up questions? So just understand the concept. Genetic mapping, complex mapping looks for more than one gene that controls a trait. Oh, and Dr. Bush asked me to stick in a plug for, in case any of you happen to want to take 103 over the summer, he'll be teaching it over the summer. So if you want to get ahead in coursework, this is a rare time when a core class actually gets offered during summer. This is a good place to jump ahead if you have the summer available and want to, or part of the summer anyway. Check it out. So May 23rd through June 10, knock out a core required class for biology majors in the span of a couple weeks. I promised you in the Blackboard posting that we were going to move away from everything that you've feared so far in class. We can stop talking about alleles, Punnett squares, DNA replication. We're going to start essentially from scratch. We're going to start fresh. We're going to talk about pedigree analysis. Still going to have to talk about alleles, but in a much different way. So if you feel like you didn't understand anything up until this point, I'm sorry, that makes me sad because that means I didn't do a very good job, but clean slate. Spring break's over. We're doing something totally different. Whole new field of genetics, pedigree analysis. So this is what I'd like to do for the rest of today and into the next few classes. So my goals are that you be able to identify this is the simplest thing, which you probably may already know how to do. Look at a pedigree and tell me, is it dominant or recessive transmission pattern? What's added on this semester for a core upper division class for you guys is predicting genotypes based on the transmission pattern of phenotypes. So this gets a little bit more in depth. And then actually conducting genetic mapping, what we just reviewed, using a pedigree and genotype information. So that'll integrate what we've talked about last class and earlier in this class with pedigree analysis. And I'm hoping, and I'm expecting, that everybody's familiar with the pedigree symbols. So that's summarized there for you to use as a reference if you want. The most basic things that you need to know are circles and squares. Guys, I don't know why we're the squares, but that's the way it works. So females are symbols, circles, males are squares. The only other thing you really need to know to get started is that when you fill in a symbol, that's an individual that's affected with whatever phenotype or trait that we're studying. Everything else we'll learn as we go. So let's see. Let me fill in this trait. I'm not going to tell you what trait it is. It doesn't really matter. We're going to try to figure out which inheritance pattern this is. And I want you to tell me why you think this is the inheritance pattern you think it is. So if you're presented with a pedigree of any organism, let's say it's human, just for fun, is this dominant or recessive? There's a particular, this is particular pattern that you look for when you diagnose dominance versus recessiveness. So why would you think it's a dominant?
The majority are affected.
Because a majority are afflicted. Let's write in genotypes. So here's the first step in pedigree analysis. Step number 1, write it down. You look for whether or not the trait you're looking for skips a generation. This helps you predict, or at least make a hypothesis about whether or not the trait is dominant or recessive. In this case, the trait shows up in every single generation. Right? We could label these P0, F1, F2 if you want, although we don't do that for human pedigrees. So if the trait is in every generation, it's usually, but not always, dominant. So if yes, it skips a generation, usually it's recessive. Here we have the trait present in every generation. So I'm going to posit, I'm going to propose that we consider this to be dominant inheritance. So step 1, does it skip a generation or not? If it does skip a generation, which we'll see next, I'll do an example of autosomal recessive next. If it skips a generation, it's usually recessive. If it's present, the trait is present in every generation, it's usually dominant inheritance. So that's step 1. Identified skipping generations or not. That lets us propose that we're going to assume that this is dominant, unless we can prove otherwise. It's like court. Innocent until proven guilty. We're going to assume it's dominant unless we can find evidence that this transmission pattern is not acting in a dominant way. And the way we would do that is the next thing we're going to do. Once you figure out if you want to think it's recessive or dominant, try plugging in some genotypes and see if all of the genotypes make sense. So if this is a dominant trait, what are these F2 filled in symbols? What are their genotypes? What would I write in right there? The genotype of an affected individual, using pluses for wild type, minuses for mutations. What are the possible genotypes of that individual? So the dominant mutation's minus. So an affected individual has to have at least one minus, right? So the minus mutation is dominant. So this individual either has 2 bad copies of the gene, they're homozygous mutant, or what's the other option? They're heterozygous, plus over minus. Everybody okay with that? So we've got a dominant mutation that causes, we're proposing that we have a dominant mutation that causes the phenotype. That's why the symbol is filled in. This individual has this disease or trait that we're trying to study. So they're either homozygous for the mutation or they have 1 copy. Either way, they get the trait. And that's true for every individual. So every filled in individual in this last generation, we don't know. We can't tell whether or not they are homozygous or heterozygous. It's the scary and exciting thing about genetics. There's so much that's unknown. But we can figure some things out. What's the genotype of the F1 dad? What are the possible genotypes? Right. Same as the kids. If you see an affected individual with a dominant disorder, it's either minus over minus or plus over minus. However, now you should be able to tell me what the F1 dad's genotype is based on what his kids look like.
Homozygous? Coincidence?
So if this F1 dad was homozygous, this genotype, what would have to be true of all of his kids? If dad has two alleles and they're both the mutant versions, what are all of his kids going to look like? They'd all inherit at least one minus from the dad. So every single kid, all 8, would have this disease because they all would have inherited a minus from their dad. So it can't be that genotype because there are some of his kids don't have the disease. So if it's dominant, this F1 father can't be homozygous. If he was homozygous for the mutation, all 8 of these kids would have their symbols filled in. There's no way to escape dad's disease. So that lets us, let's see, what's the F1 mom's genotype? Yeah, has to be plus plus because we're talking about a dominant mutation. If she had at least one minus, then her symbol would be filled in as well. So now you should be able to tell me all of the genotypes of all of the F2 kids. Are the filled in symbols minus minus or minus plus? Is there any way for any of those F2 kids to be minus over minus? No, because we already figured out that mom has to be plus over plus. She doesn't have any minus to contribute to her kids. So the genotypes, if this is being transmitted in an autosomal dominant pattern, those are the genotypes of those individuals. We work it out based on